We are entering a new era where there's a cure for rare genetic diseases, creating medicines tailored to a person's DNA, and screening for conditions before they even start to show signs. For the first time in history, scientists can read, edit, and even rewrite DNA. And that is only because of the $3 billion Human Genome Project that gave them the entire genome. But what is a genome and what does it really hold? Imagine the nucleus of every cell in your body is like a vault. Inside this vault are chromosomes, and we have 23 pairs of them, each containing a segment of the whole DNA packed with millions of base pairs. And where exactly is a gene located? That's called a locus. It's more like the address for a gene. Think of a gene like a single sentence in a paragraph in a vast book. For each gene, we inherit two versions, one from the mother and one from the father. These versions might have small differences called alleles, and these tiny changes are what makes us who we are. So, what does this DNA do? The coding regions in the DNA hold instructions for proteins, the little machines that keep us alive and functioning. And the non coding regions instruct gene expression, where certain cells use only a particular gene and silence all other that don't contribute to their function. Did you notice how it all started from the DNA and ended with proteins and other functional molecules? This is a rule. And back in 1970, Francis Crick came up with a groundbreaking theory, the central dogma of molecular biology. He said that the genetic information flows only in one direction, from DNA to RNA to protein. And if any cell broke it, and somehow proteins could rewrite DNA, everything we knew about molecular biology would change. The DNA does more than just create proteins. Its most critical role is passing down traits and susceptibilities to diseases, shaping who we are and how we respond. The genotype, which is a genetic code, leads to the phenotype, the traits we see and feel. And understanding that link became the future of genomic research and healthcare. But to understand the code, there had to be a way to look at it, not segment by segment, but the whole genome as it is. In 1990, a bold initiative was launched with the backing of the NIH and the Department of Energy, a project to map all 3.2 billion base pairs of the human genome and a few selected non-human organisms. It was called the Human Genome Project and it came with a $3 billion price tag. But even before it began, the project faced intense criticism. Many scientists doubted its potential, expressing through multiple letters stating various reasons. Most of the sequencing work will not be creative or innovative, and graduates do not wish to spend their graduate or postdoctoral time in the repetitive process. 95% of the DNA is junk, it's just fillers. We'd rather start from an important loci. The big science project is not worth it, as it would take close to $3 billion. Starting from a useful segment would be a sensible beginning, a letter from Nobel laureate Christian Anfinsen himself, who laid the foundations for the computational models for protein sequencing we use today. Despite these concerns, the project was approved, becoming the largest coordinated biological research effort in history and being publicly funded. The researchers recruited donors through public advertisement and with informed consent collected blood samples from them, from which they extracted the DNA. But how do you sequence something as vast as a genome? That's when they turned to the technique developed by Frederick Sanger, the Sanger sequencing method. Imagine you're writing a long sentence, but you've randomly placed special stop signs. Every time your pen hits one, it stops writing. Sanger sequencing works the same way. These special stop molecules called dideoxynucleotide triphosphates randomly halt DNA replication at specific points. And by collecting all the fragments and lining them up by size, scientists can read the DNA sequence letter by letter. Using this technique, they sequence simpler genome first, such as baker's yeast, a flowering plant, a worm, and a fruit fly. And once these were successful, the human genome sequencing began. But the traditional Sanger method was painstakingly slow, labor-intensive, and not scalable for something as massive as the entire human genome. And that's when a faster and efficient technique called the shotgun sequencing was developed. Instead of sequencing the genome in a long, continuous strand, the DNA was first randomly broken into millions of smaller fragments. Each of these fragments was then sequenced individually, and powerful computational algorithms were used to analyze them and piece them back together. This approach allowed scientists to massively scale up sequencing projects, making it feasible to tackle complex organisms like humans, with a staggering 3.2 billion base pairs. In April 2003, the Human Genome Project Consortium announced the completion of a nearly complete human genome sequence. This version marked a major improvement over the earlier draft, which had over 150,000 unresolved regions as gaps. This new sequence covered approximately 92% of the human genome with fewer than 400 remaining gaps. And on March 31, 2022, the Telomere to Telomere Consortium announced that with every last base pair accounted for, the sequencing was complete, and it was a gapless assembly of the human genome. The human genome sequence generated by the project was actually a patchwork of multiple people. However, the majority of the sequence came from one person of blended ancestry. 70% of the original reference genome sequence was generated from that individual's DNA, and with the remaining 30% coming from a combination of 19 other individuals. So, why did all this matter? For the first time, we had a complete reference genome, a blueprint of human biology stitched together. It gave us more than just a list of genes. It brought down the cost of genomic sequencing significantly. It sparked the beginning of systems biology, the idea that health and disease are about interactions, which led to the launch of ENCODE, a massive NIH initiative to figure out functional parts of the genome, and that changed the domain of proteomics. But even with the entire genome in our hands, a critical question remained. Now that we know what's common, what about the differences? And that question led to the next revolution. While every one of us shares 99.5% similar DNA, the 0.5% variation occurs from the modifications in either a small section or a large scale. Variations in the DNA sequences are what makes every one of us unique and also affect how we develop diseases and respond to other agents. Inside our DNA, small variations quietly shape who we are. Some are tiny single nucleotide polymorphisms, where a single letter in the genetic code is swapped, much like a typo. Most SNPs are harmless, but when they land in sensitive regions, they change everything from protein synthesis to cell regulation. Other variations are much larger, like copy number variations, where thousands or even millions of bases are duplicated or deleted. Together, these small and large variations weave subtle differences into the human genome, differences that can influence health, disease, and evolution itself. The Human Genome Project gave us the complete blueprint, the reference genetic map to the human body, and for the first time in history, we had the full DNA. But having the map alone does not tell us where the problems are, and that's when the HapMap project was initiated. Think of the HapMap as placing pins on the genome, markers that highlight regions with common genetic variations. These markers help identify patterns of genetic inheritance and variations. HapMap significantly reduced the effort for genome-wide studies, making it easier to sequence and find association with genes. It changed the way diseases were looked at, and as mentioned in this paper, after HapMap, GWAS became as easy as expected and intended. To perform a GWAS, we just collect DNA from patients, genotype it using commercial arrays, and do a bit of statistics for the results. And as computers became faster and more affordable, scientists could start handling genomic data in a way that were never possible before. Sequencing moved from being a slow step-by-step task to a massively parallel process. With the advancements in computing platforms, tools like Parabricks, which is built to run on GPU clusters, take traditional genomic analysis pipelines and supercharge them. A 5-hour whole genome sequencing task that was performed on an Illumina system can now be completed under 5 minutes by coupling them with these platforms. And today, we have technologies like CRISPR, which allow gene editing to cure diseases and inherited errors, cancer genomics for targeted cancer therapeutics, and precision medicine, which are treatment plans tailored to one's biology. With generative AI, we're stepping into the future of digital twins, where parts of the healthcare will be virtualized and every scenario will be first tested out on the virtual twin, thereby reducing risk significantly and improving the quality and outcome of patient care. None of these breakthroughs would have been possible without the understanding of the genome, and that was made possible only because of the $3 billion Human Genome Project.